Okay, hello. Welcome to another session of the Friday Scientific Seminars at the Museum of Natural Sciences. It is my pleasure to welcome today Sonya Clegg from Oxford University. Sonya she's from Australia, and she did her PhD at the University of Queensland. And then she did postdocs at San Francisco State University. That's where we met many years ago. And then she's been at Imperial College in London, and now she's been a professor at Oxford for the last 10 years. And well, Sonya has done a lot of very exciting pioneering work on what happens on islands upon colonization, and with things like founder effects and selection and the role of population size, and working with species that are called great speciators, as she will explain, like in this case bird species that are very good at colonizing islands and then very good also at becoming new species. So it's a pleasure to have you here, and afterwards we'll go and have a beer outside if you want to talk to her with more time. But we'll have time for questions, hopefully. So take it away.
Thank you, Borja. And okay, is that good? Yes. Okay, thank you. Thanks for coming out, everyone, on a Friday afternoon, and for coming and for the invitation, Borja. It's been a long-run thing. We've been trying to arrange to come at some point, so it's a pleasure to be here. So this afternoon, we're going on a journey to the South Pacific, where I've done a lot of my work, and I've worked on lots of different systems, mainly birds, but I keep coming back to this system of this great speciator system in the South Pacific. It's really good for answering the kinds of questions that I'm interested in. So this is one of, I call it my island. It's not really my, it's an island on the southern Great Barrier Reef where we have a long-term colour ringing population of silvereyes that has been going since the 1960s. So it's a rather nice place to do field work. And then this is the beast in question, the silvereye, which you'll get lots of pictures of. So I'm kind of going to start where some people finish, and a lot of the work I'm presenting today comes from two of my PhD students who've completed, and a new PhD student, Abby Williams, and I really want to acknowledge them up front because they're the ones who really crank that bioinformatic machine for me, because it does get beyond you at some point in your career. Okay, so to start with, I want to talk about a couple of, well, they're really old ideas in island biogeography and island evolution, just to show where this work sits within those broader ideas. And many of you have probably heard of the taxon cycle. An idea proposed by E.O. Wilson that species go through phases during their kind of lifetime, if you like. And it starts, so this is Jared Diamond and Ernst Mayr developed this idea. So starting with this idea that you have supertramps and tramps, these species in these archipelago island situations that are really good colonizers, and they and so each of these is an island, it's just a cartoon, and they colonize, but they kind of stick at the peripheries. If it's a small island, maybe they get into the center. But so the idea, they're not very good competitors, but they're really good dispersers. And then it goes into this second stage that they call, gone too far, the great speciator stage. And that's the phase I'm going to talk about. And the term great speciator has become quite popular again. You'll see it in lots of titles of papers. And it's become one of what I call a concept cluster. People are using it to mean different things. Some people call a great speciator a group that has a really high speciation rate, for instance. But in its original incarnation, it was actually used to describe a species that had a wide geographic distribution, like on many islands, but that had high sub-specific diversity. Okay, so it's still looking within one species. And that's the way, the original description and how I am interpreting it for this talk. So what happens is the geographic range widens and the population differentiation starts to increase. But then as the taxon cycle progresses, it goes through these other phases of specialization and speciation. So things becoming different, maybe contracting to the middle of islands, and then relictualization, some extinction. So if you go too far down this route, eventually you can't kind of go back to the start. But there are these little loops that can occur at the start. So you can have some divergence, a bit of divergence, morphological, genetic, and then they might go into a dispersal phase again. So it's these rounds of dispersal, colonisation, differentiation that form the great speciator stage. Sorry, I have to do it there. And the idea was, the great speciator idea was really promoted by Jared Diamond and Ernst Mayr. And they developed it from looking at bird subspecies across northern Melanesia. So this is north of where I work, but so northern Melanesia is up here. I'm going to be describing work from southern Melanesia. And they made this fairly arbitrary cutoff that if within that region, if you had more than 7 subspecies, or you had 4 to 6 subspecies and your subspecies to island ratio is more than 0.33, you could be called a great speciator. So not a very transferable concept beyond the region. And they identified 31 bird species that had this broad geographic range but high morphological diversity. And they called it a paradox because the idea that for many speciation models, at least you need some phase of allopatry, some reduced gene flow. But if you're really good at colonising a wide range of areas, how do you simultaneously get broad distribution and differentiation? So it's called the paradox of the great speciators. So this is just a slight aside, something I'm working on still with my former student, Andrea Estandia, is to try to make this concept more broadly applicable and have, we're working on this great speciator index. And in the end, it turned out to be quite simple, but Andrea's done this for all bird species on all islands all over the world, and she's ranked them, and now she's looking to see if we can describe variation according to these. So this is not complete work, but what I wanted to show you here is that's the island thrush, if anyone knows that. That's number 1. So a lot of these top group are in northern Melanesia, but you also get things like warblers and things in the Americas. So it's a concept that can be extended, and then maybe it will be actually a bit more useful. Oops, sorry, I've got to get this right. So I said it's a paradox, and the resolution to the paradox that people have said is there just must be a change in dispersal post-colonisation, and it's probably quite rapid. People don't know what the mechanism of that change is. There's multiple things it could be. Behavioural flightlessness is one. They could still fly, they just choose not to, all the way through to ultimately changes in morphology, physiology that might be underlaid by genes or plasticity or all the things that happen between genes and phenotype, like epigenetics and development. So a lot of the work on great speciators, it's an island concept, and the work has been done on islands. And the problem with that is we don't know what these birds would do if they were in a continental situation, right? Would they still be a great speciator, or would something change? Does the geographic context matter? Does it have to be on islands? And so that's something I'm going to address today. So change in dispersal ability or capacity is one component of another big area that I work in, which is the island syndrome, this suite of morphological, behavioural, physiological, life history changes that tend to occur on islands. And another PhD student recently did a review of what this looks like in birds. So we definitely have seem to have reduced dispersal, but we have other behavioural changes like widened niche. This is the vampire finch, definitely doing something there that's not very common. We have the island rule. So this is where if you start small, you tend to get large. If you start large, you tend to get small. For birds, there's lots of other changes, more rounded wings, which is linked to dispersal, lower sexual dichromatism, and this shift to a graviportal body plan, which is also linked to dispersal. So that is something like an emu would be cursorial, and something like a dodo would be graviportal. There's all these life history changes too that occur. I'm not really talking about these components today, and of course the behavioural stuff, but we're working on all aspects of this. So what I do want to talk about today, apart from dispersal, is the island rule, and how can we explain repeated patterns of phenotypic change across islands that are really quite different. They can be across wide latitudinal ranges, they can be different levels of isolation, and so can we understand that within a group in terms of them recycling standing genetic variation, or what is the role of de novo mutations in contributing to these kind of repeated patterns that we see as well? Okay, so what I want to do is take these old ideas about stage 2 of the taxon cycle, the great speciator stage, the ideas of the repeated patterns that happen of reduced dispersal and change in body size, and try to apply some modern genomic methods to see if we can tease out some of these things. So first of all, with the great speciators, it was set up as a paradox, and they said, oh, well, the solution is a change in dispersal. However, if we're only looking at things in the island situation, like I said, we don't know what happens on a larger kind of continental mass. And if we look at the speciation literature, they find that the groups that have the highest diversity of species, the highest speciation rates, tend to be things with medium dispersal ability, right? So not so much as to cause, completely homogenizing gene flow, and not so little as to reduce the opportunity to move into new niches. So perhaps these birds that we're looking at, they just have some kind of medium level of dispersal, and it's just right to get that distribution, divergence kind of balance right, so you become a great speciator. So maybe it doesn't involve specifically in a change in dispersal capacity. But following on from that, if we do have a change in dispersal, is there any genetic signature of that? The people who work on migration have found some nice switches, like to switch things on from migrant to non-migrant, and we're seeing things change with climate change, that things are no longer migrating in some areas. So for my system, can we find that? And then related to the island syndrome, the dispersal is part of that, but thinking about this repeated, my birds were small, so they became larger on islands, and I'll show you some data for that in a bit. But do they just reshuffle this standing genetic variation, or do they get their novel mutations also contribute? I mean, for any kind of repeated evolutionary pattern, we know we have to rely on reshuffling standard genetic variation, because it just takes too long for mutations and the right mutations to arise, but do they play any part in some systems? And then I want to finish with something that I hadn't thought about very much, but when you start working on birds that colonise islands really frequently over evolutionary time, and then they speciate and there's another wave, what you end up with lots of cases of secondary sympatry. And so it's kind of this, range expansion pulses that happen in the taxon cycle, and that leaves opportunities for introgressive hybridization. My group of birds, people said, oh, they don't really hybridize very readily, so I just believed that for a long time until I didn't, and now I'll show you why. So to introduce the species of the Zosteropidae, I know Borja works on these as well, but so this is the worldwide distribution, including the orange, but this is the one species that I work on called the silvereye, and I work on other species in this area as well. So it's got continental distributions, it's got insular distributions, it's got lots of cases of secondary sympatry, sometimes a triplet on one little tiny island, a big one, a medium one, a little one. And yeah, so of course, all of you would know that in each of these pairs, or each of these pictures, that's the silvereye, that's the silvereye, and that's the silvereye. Did you get that right, Borja? I hope so. Okay. One of the nice things about this system is that this bird has colonised islands in the southwest Pacific over a range of timescales. Some of them are very old, like these ones up in New Caledonia and Vanuatu, 100,000 plus. This one we think is a bit younger. The island's only been there for 4,000 years. That's the island I showed you in the opening slide. Another old one here. Then in the 1830s, we have this historically recorded sequential colonisation, all natural, of this part anyway, and it was all really finely dated. And so in Norfolk Island, it arrived in 1904. Someone was there, collected some, and said, what's this bird? It's different to the other two white-eyes that occurred there. And it was also taken to, this was a human-mediated introduction to French Polynesia, to Tahiti, after which the populations built up and it started to island hop out there as well. And that's nicely dated. So we have this spectrum of timeframes over which to look for morphological and genomic changes. So I spent a good chunk of my time island hopping after these birds, and it was amazing work and really, really hard work at times. That's not a cloud, that's a volcano going off. So yeah, it was by foot, by canoe, by very small plane. So I've actually been out collecting blood samples for a couple of decades, but I don't do that work so much anymore. And what I'm going to present relies on a huge number of people who've helped me, local communities and different students and collaborators over the years. Okay, so what is the silvereye a great speciator? So when I was thinking about this, people write down, it's got however many subspecies, and with 16 or 17 morphological subspecies, you can argue about that. 11 on islands, 10 exclusively on islands, and that's some of the different forms. Some have a black head, some don't. That kind of falls within whichever great speciator idea that you want to use. But because there was also this recent, last couple of 100 years, another flush of colonisation, I call it a trampy great speciator. So it's parts of it are like a super tramp and parts of it have come into this great speciator stage. Or most of it's a great speciator stage, but it's got looping back on this super tramp idea. So for the first question about, so it forms lots of subspecies on islands. What does it do on the continent? So it's a strong system to do these comparisons because we have all these continental ones, and you could do this probably for Africa and the islands in the Indian Ocean as well. And so Andrea did this work. It's published 2 days ago in Molecular Ecology, which is nice. So whole genome sequences. We didn't quite get all the subspecies, but we got quite a lot. And some other wide-eye species in there as well as Borjas bobonicus here as an outgroup. And so first of all, setting up what the phylogeny is, this is all the sampling that we did. And you can see all of these island populations here coming around, and then we get into the continental stuff here, and the very recent colonisation. And so it seems that if you put this bird in an island situation, it's going to start to diverge more. And it's, if we look at, water barriers, this is a really good coloniser. So are water barriers effective at some point for this species? So if we look at FST, so just a measure of genetic divergence among islands, so the higher you are here, the more genetic divergence you have, versus all these continental subspecies. It's a wide range, but it's relatively low. So water barriers are effective eventually in getting this kind of divergence. On the continent, we expected some isolation by distance, but they just keep moving. And there are biogeographic barriers for many passerines in the Australian continent, and they ignore them and they go across them. They also, they are actually not afraid of going large distances and not afraid of going over water. So they don't have a predisposed reluctance to cross water. So it's not like they have some kind of medium dispersal to start with and it stays medium on the islands. They really do go. These are band returns, ringing returns in Australia. There's this one's partially migrant, it crosses Bass Strait. We know, they've crossed this big stretch of ocean here in the last 200 years. We get vagrants on all the little islands up and down here. So they freely initially disperse across water, but once they get there, we do think they change. So this is telling us that there is some change in dispersal. It's not the kind of null model of some medium dispersal capacity that's just in the right zone to generate a great speciator concept. So, Mayer and Diamond, they're right. It's some kind of, for this one at least, reduced dispersal. But of course, we could, I'll just go back. This could be because of founder events and, squeezing through bottlenecks as you colonise. And I've done quite a lot of work on following that sequential colonisation, what happens when, and because we know how long they've been there for. So this is from Tasmania to New Zealand and outlying islands, and then Tahiti. And so what we were doing was saying, how much diversity do you lose in a single step? How much do you lose in 2 steps, 3 steps? And so as the number of founding steps increases, your FST, you can get it up there, but any single founding event doesn't do very much. These things colonise in flocks and they recover very rapidly from small population sizes. So genetically, there are not huge effects. And they certainly don't get to the divergence or the loss of diversity that we see. These are older island colonisations of thousands and hundreds of thousands of years old. And this is tracing the fates of particular alleles. This is all single digest rad sequencing, so not whole genome now. But what we find is that, that erosion of diversity is just the loss of alleles that are rare in the first place. But I kind of like this graph because it's something you can show first-year students that there's stochasticity in something that can start low, can also end high, but probably more rarely than it just goes extinct. Okay, so, and just also an aside, this is the human-mediated introduction to French Polynesia, was done by this very interesting man called Easton Guild, who was an aviculturalist extraordinaire, introduced thousands of individuals of nearly 50 different bird species for no apparent reason, and silvereye is one that took. So there's a whole lot of work to do there on a really nicely dated system. He's very interesting to read about. Okay, so by comparing continent and island, we do see that in the island context, silvereyes are doing kind of the great speciator thing. We don't think it's due to bottlenecks. We wanted to know if there were any, underlying genetic switch to some of this. And actually this work was published a couple of years ago, this was before we had many whole genomes, and so we decided to do some candidate genes. And we looked at 6 candidate genes that have been implicated in changes in migration or kind of dispersal, personality things. Even in humans, DRD4 is one that, the out of Africa hypothesis that, people had this propensity to move. So there are these candidate genes, and this is just results for 4 of them. So these are all microsatellite candidate genes. So the difference in allele length is up here. And these are for a whole lot of different populations. And they are coded according to two large genetic divergent groupings, so these ones and then all the black ones out in southern Melanesia. And some of them don't vary much at all. We can't really go very far with that one. This one varies, but not in any pattern that we could see was obvious. However, this one, CREB1, has shorter microsatellite profiles for populations that don't disperse. And it's not explained purely by phylogenetics, because two of the ones that occur in this kind of clade who don't disperse have this lower, this shorter microsatellite genotype. So these you can go and then see what these genes do. They do a whole range of things, but a lot of them are doing, like, so clock is to do with circadian rhythms, and these ones are a whole lot of physiology as well. So clock doesn't look like it's doing much, but if you look at these yellow-coloured dots here, these are individual birds who we know were migrants. And so we thought, oh, it looks like they might be doing something a bit different to individuals that we know are not migrating. And we were able to do this in, so this is just showing the mean CREB length, again, with the two genetic groupings, showing that there are two non-dispersal kind of phenotypes that belong to that genetic grouping but fall within here. And then we were able to go in Tasmania. I said they're a partial migrant, so they self-sort for us. If you go in winter to Tasmania, they're the birds that stay, and you jump across to continental Australia and catch that subspecies there, they're the migrants. And so when we looked at this, there is a overlapping, but a mean difference in that the migrants tend to have these longer allele lengths. And this is the clock gene that's related to circadian rhythm. So it's very hard when you're working on a non-model species to kind of link genotypes and phenotypes. But so what we're doing next here is actually we went back and did a proper sample, and we're just now doing the whole genome sequence, and we'll kind of do some GWAS work on that. Okay. So that's the dispersal stuff. So thinking about body size. When these birds go to islands, they get bigger in general. There's a bit of wobbling around, and remember, this is over these are really young, these are really old. Once you've been out on the islands for a long time, so we have, these are mean, so there's 3 bumps here because it's quite variable within those, within that southern Melanesian region. But essentially, you're tracking to get larger. And by larger, so this is on Heron Island. It's only 80 kilometres off the coast, a different subspecies. Mainland vagrants sometimes arrive, and we're able to get these photos. And that's about a 40% body size difference in something that we think only evolved some thousands of years ago. And behaviourally, that makes a big difference. And I've actually seen these birds kill one of these birds because the resources are limited and they're not sharing. So there's a lot of, there's huge body size differences, and that one is, so you know where you are, 4,000. So that's this one here compared to like this one here. Okay, so we have this directional shift in body size. And so what we wanted to do was do some genome-wide association studies on this to find out if there was any kind of outliers associated with body size. And everyone said we were a bit mad because it's a polygenic trait, right? Body size is very polygenic in many cases. So the first thing we did was, we had to rebuild our phylogenies, and so when you're doing these genome-wide association studies where you have a trait and you're trying to match it to something at the genomic level, you need to correct for population structure. So that's what we were doing there. So we had a reasonable sample size, I think, for a natural system. And what we found was, and so the question here is, is the repeated body size evolution, is it just recycling or reshuffling of standing genetic variation, or do we have any putatively novel mutations that also contribute? So the top graph here, so it's all the chromosomes lined up, and then this is how associated all the different variants are. And so that's, I can't remember exactly what that dotted line is. If it's, it'll be some Bonferroni thing saying above that is highly associated, but there's some here that are quite well associated as well. And so this is with body size, and then we looked just at bill size as well, where there was a little less. And then we went and looked for, a whole lot of the kind of top 1%, I think it was, and said, so they're associated with large body size. Geographically, where do they occur? And they kind of occur everywhere. So this means that they are using pre-existing genetic variation. They're reshuffling it as they're going to new places, which is how you would expect a repeated evolutionary pattern to occur, because it's just easier to do. But we did find this one outlier here that was restricted to just one set of populations in southern Melanesia. And so what we did then was we kind of focused in on it to look at this peak, and found the point mutation that was associated with it. And then looked at what body sizes they were. So if you're a CC, a homozygote CC, you can have a wide range of body sizes, but if you are a heterozygote with a T or a homozygote double T, you get bigger and bigger. And these only occur up in this region here, and not down here. So we submitted all this, and the reviewers came back with, but there's so much population structure there, it might be to do with that, which is fair enough. And so what we said was, okay, we will zoom in just on that northern part of Vanuatu now, where we have lots of variation but lots of gene flow moving around, and we'll go and see, you get a reduced sample size now, but there's still a peak there, and it is still associated with larger body size within that reduced area. So we're pretty confident that this is so it's not widespread across all silvereyes. It seems to have evolved there sometime about 8,000 or 9,000 years ago. So we're calling it a putative novel mutation that contributes to large body size, but that is not the main way that silvereyes achieve large body size. But if you're lucky enough to get one, you can be super big, like they are out on those islands. And the next thing, though, you can do to kind of investigate it a little bit further, we wanted to know, does this represent something that swept through the population that does look like it was really advantageous? So has it undergone a selective sweep, a hard selective sweep? And there are various ways to do this, but one thing you can do is build phylogenies for that particular area. And so this one is taking that point and some area around it and kind of building the phylogeny. And what you see is the pinky region here is all up in Vanuatu, and this kind of long branch here suggests that that's consistent with it kind of sweeping through. It also has really high FST compared to the flanking regions, a pattern you would also expect from a hard selective sweep, where just one mutation arises and sweeps through the population. And we can compare that to the second highest outlier on a different chromosome. And you get this kind of, you can build a different kind of tree, and it's kind of all messy and everything, but kind of what you would expect of something that's just popping up or being reused in all these different populations. And you get a very different FST profile as well, where instead of a hump, you get a U shape. And this is what you would expect when you have the focal gene segregating on lots of different genetic backgrounds, okay, across the entire region. Okay, so, and then you can go and do various tests for selective sweeps. This is kind of the composite test where we can look for this peak here, and it's actually composed of all these different, they kind of roll all these together. And so this region is doing everything that we expect from having this negative, I won't go into these statistics because I don't have time, but negative to G minus D. It has low genetic diversity, as you would expect. If something is not, something so favoured, it sticks to what it is and it sweeps through the population. And it also has, the region around it has a higher linkage disequilibrium value than points on other, outlying points on other chromosomes that are more shared across populations. So our current status is that reshuffling standard genetic variation gets you a long way towards your increased body size. Occasionally, if you get a new mutation like this, and there are, we do a lot of ecological work as well to show what the advantage of large body size is, and I'm not showing that work today, I'm happy to talk to people. But as I mentioned, that big bird killed the little bird. It's good to be big if you're in these high-density island situations. But then it occurred to us there's another place that this might not be a de novo mutation. There's another way to get genetic variation in these systems where you have secondary sympatry and you have these really large wide-eye forms out there, is that you might hybridize and there might be adaptive introgressive hybridization. So I showed you some of these pictures before. Cases of secondary sympatry. So now we've done all the phylogeny, so we know who got there when and when they diverged. So there's, and these are replicated. So this one across Vanuatu is a pair that, I don't know how many islands, like probably 20. Grande Terre, New Caledonia, there's a couple. This is the one in Lifu in New Caledonia where there's 3. And then Norfolk Island, which I just want to talk about a bit to finish up. So this is the one that arrived in 1904 from New Zealand. This is an endemic white-eye that was already there. And there was an even bigger one that was extant at the time when they arrived and when the silvereye arrived. So that's the silvereye in each case. So I've done a bit of work on Norfolk Island, and when we were looking at the sequential colonisation of the silvereye, it occurred to me that the Norfolk Island silvereye was a bit too different compared to what all the other ones in New Zealand and even French Polynesia that did go from, because it was human-mediated introduction, it was just a handful of individuals, where these are flocks of birds arriving. And its genetic profile, and I should have put a picture up, it was just struck me always as being too different. And so what happened on Norfolk Island, so pre-1904, we had 2 endemics, this big guy and this slightly smaller one. The silvereye arrives. It takes off. By 1913, it was the most common bird on the island. So demographically, these things are, changing the whole scene. This one just declined. By the 1980s, it was gone. I don't think they've actually called it yet, but it must be any day now. We must be pushing past the 50 years soon. And then there were only 2 left. And in the 60s, there was a publication about some specimens held at the American Museum of Natural History that described them as hybrids between this one and the colonist, the silvereye. And so I thought, well, maybe this silvereye now is so different because it hybridized, but I didn't see any evidence of that on different field trips. So what we've done is I went and sampled. I sampled these in 1996, a lot more than that, but that's what we have whole genome data from. So then we went to the American Museum of Natural History, and we were able to get some specimens of the one that's gone extinct before 1904, the one that is still extant, but so we don't think this has had opportunity yet for any introgressive hybridization, so we're counting that as our kind of pure slender-billed white-eye. And then we were able to get the 3 purported hybrids. a smattering of them from 1926, and we just did whole genome work on this to try to unravel what's happened over this decade or more of secondary sympatry. And these are the specimens in Tring, so you can see the size difference in real time. So what does it show? They were hybrids. So here's the silvereye, here's the extant island endemic. They sit nicely between the two. They accidentally gave us another hybrid. It wasn't identified as a hybrid, but it's a hybrid between the silvereye and the one that's gone extinct, okay? So there's the opportunity then for ghost introgression, where something that's gone extinct in the wild has still genomic material represented in an extant species, like Neanderthals in us. And so this is just the admixture plot where you can see the extinct endemic, the one they called lateralis, but is a silvereye, but is actually a hybrid with maybe a little bit of something else in there, the three obvious hybrids. And then these blocks here are the mix of contemporary and old Tenurostrus. So then we were wondering, you know, what are these birds? And so by looking at the coverage, so in birds, the female is the heterogametic sex, so ZW. So we matched up to the W chromosome to see if there was any there. And all these these four that identified hybrids, they're all male. That's what you would expect for a Haldane's rule, right? So if two species breed, and if one of the offspring sex is rarer, infertile, or absent, it tends to be the heterogametic sex. It's a small sample size, but it's following that we would expect them to be males. But then, and we wanted to know who the parents were, so we extracted the whole mitochondrial DNA, so the mitochondrial DNA transferred from the maternal line, and you can see that, so the orange are those three, and then that's the green one, and they all fall within the silvereye maternal lineage. So the mother of these birds was the coloniser, and she came there, and there were these amazing, large, very apparently attractive males, and formed hybrids. I will say this is all this last couple of weeks we've been doing this work. And then we wanted to delve a bit more and just say, well, okay, they're in the museum, but are they still hybridizing? And so we did this thing called ABBA-BABA test. I'm just quickly going to describe what they are. So if you have, so the grey is like what a species tree would be. The lines are single gene trees. If every single gene, bifurcates as the species do, you would end up with this pattern that's called BBAA. But that doesn't always happen. We have incomplete lineage sorting, and so you might have patterns like BABA or ABBA. And that can occur, this ABBA pattern in particular can occur, it could just be from incomplete lineage sorting, okay? That not every point in the genome has sorted itself out by species. But the other way it can happen is if you have hybridization later on, okay? And so what we're looking for is, you would expect an equal number of these just by incomplete lineage sorting. So we're looking for a little extra ABBA that would indicate hybridization with this other form. And when we looked at this in these three, the answer is yes, is the short answer. So we substituted this is the extant endemic, the extinct one. So we looked for hybridization from there into there and from there into there. So this D statistic, we have this little bit of excess ABBA in both cases is significant, telling us that the silvereye has taken on genes from both of those endemic forms. And then we can look across the entire genome with all the chromosomes lined up here and look at where it has actually introgressed. So it's been pretty genome-wide. And the next thing you could do is go and say, well, what's that point? What's that point? We decided to kind of group it into kind of the functions of different genes and to see which ones were enriched, to see if it's got any adaptive value. And literally this came this week. And so there's a program I hadn't used before. And well, I still haven't used it because my student used it. But basically what it's looking across those regions and grouping those outliers into different kinds of biological processes. So this is growth, skeletal formation, all that kind of thing. Molecular functions is how things move across membranes. And the human phenotype ones are phenotypes that we know from humans have, like in most of these cases, it's something to do with the jaw and the mouth, and so there's something going on there, we think. So we've got to investigate this a lot more, so we could build a story that some of them could be adaptive. So I think secondary sympatry is probably going to be fairly important for this whole story as we keep going. Okay, so just to wrap up, the great speciator concept keeps getting used widely. We do think, at least this is one case where we've been able to show it is a change in dispersal capacity rather than a maintenance of intermediate dispersal capacity. We have some candidate genes to show that there could be an underlying quick genetic basis to be able to switch off this dispersal propensity. Along with that, we get this increase in body size that comes about from reshuffling of standing genetic variation. And if you're lucky, maybe a de novo mutation, or that could come from hybridization with one of these large white eyes out there, and we're going to look more into that. And with that, I'll leave you a cuddling silvereye pair. They're monogamous, so they're very sweet. And thank all these people and you for your attention on a Friday afternoon. Thank you.
Thank you. Sonya, any questions from the public? I'll start with one. Do you have enough within island sampling in some of the larger islands to see if there has been, since dispersal has been so restricted, if there's been any degree of structure within islands?
The answer is probably no, but I suspect like within these really large islands like New Zealand, North and South Island, there'll be maybe some microstructure there, but it's all been, that's for the recent stuff, it's probably too recent. The other islands, yeah, I do have a few variable sites, but yeah, none of the larger islands are really densely sampled because we were trying to, the trade-off is to get to as many islands as possible.
Right,
yeah.
definitely.
But it could be.
Hi, thanks a lot for a great talk. I was interested to see that the great speciator concept seems to revolve mostly around phenotypic variation, which doesn't really necessarily relate to reproductive isolation. And I'm wondering whether you have a sense that if this actually leads to speciation in the long term, or like what's the dynamic around that?
For my system, I'm starting to think that there could be quite a lot of interbreeding, but I think, yeah, so the original concept is on morphological subspecies, but we can now go and get the genomic data to say which are valid and not. Whether those different forms proceed along the speciation continuum or not will depend on a number of factors, and I think isolation is kind of key, and a long period of isolation. They all look different to me, but I'm sure to some of you they all look like the same bird, right? So I have got a new PhD student, and we're working on song, so we're going to do a lot of playback and of all the different subspecies to determine which ones that they would respond to and or recognise even as their own species. So that might give us more of a a closer kind of ecological feel for reproductive isolation, which if you put these things together in the right context, I've decided they will. From a genomic point of view, their genomes will go together. So it's going to be some behavioral thing keeping them apart.
Okay, right, thanks. And when they occur sympatrically, is there assortative mating based on like body size or other traits?
Well, I mean, the secondary sympatry is between different species of white-eye that are 1 million years separated. So we don't observe cross-species matings. I'm not there usually long enough. We've just got this trickle of intraggressive hybridization to tell us that it sometimes happens. So when the idea on the secondary sympatry is when something first arrives somewhere, it's usually in small number, and there's something called the desperation hypothesis, that they're so desperate to find a mate, which may be the case with these female silvereyes, that they choose a local male white eye. But it could be choosing on body size, it could be choosing on song, or it could be just there's no one else here, so which is the idea. And then the idea is that it declines over time, that it's really transient. It's like, let's test this, oh no. But what we've, at least over 100 years, which is a blink of an eye in evolutionary time, it's still happening. But what we want to do is go and look at these ones that have been secondary sympatry for 100,000 years and see, are they still, or are they hybridizing?
Thank you for this very beautiful and interesting talk. It's super interesting, and I was wondering if you explore anything related to chromosomal inversions. It's complicated. There's some structural variation that we're very interested in that is potentially a quicker way to cause reproductive incompatibility, right? And so we haven't done long read sequencing yet, and that's something I would like to do, but just to confirm that they are inversions. But yeah, there are definitely some interesting inversions, especially between the Southern Melanesia group and that kind of Australian group. I haven't done that work before, so I'm very happy to talk to anyone who can then guide me on what the next step is. But yeah, it's there, and I think it's interesting. Thank you.
Yeah, thanks for this great talk. I'm very happy I was here by chance. And okay, now I just have a curiosity. I know that there are white eyes in Hawaii.
Yes.
I don't know if, well, as far as I know, they were introduced, but is it possible that they arrived by dispersal?
No, they're Japanese white eyes, and they were introduced.
Okay.
Yeah.
And have they speciated there, or has anybody?
They haven't speciated, but they will be divergent in some way. I haven't actually seen anyone who's done that work. I mean, they might be doing that. I think people study them more from the point of view of avian malaria, because they're a terrible reservoir of avian malaria for the endemic Hawaiian fauna. But, it doesn't take long for morphological changes to start to happen. And I didn't present it today, but in that 200-year kind of island hopping thing, I was able to go and look at how much morphological change has happened. We were able to model the founder effective population size and say, well, how much morphological could you get by chance, given that effective population size? And the change that we see is too big to just be chance. So we know they get to a new place, selection starts to operate, they start to get bigger. I don't know about the Japanese white eyes in Hawaii. Maybe they have, I don't know. Yeah, it'd be interesting to look.
Thanks.
Yeah, thanks for the talk. In a more general context, I'm talking about the changes in dispersal capacity. Have you considered that in geological times, islands might be connected and disconnected through changes in sea levels? And then you don't need to change your dispersal capacity, but just wait that the connections start?
Yeah, you should definitely take this kind of dynamic island formation into account, but these are oceanic islands that have not been connected.
Well, depends. I work on Cape Verde, which are ocean islands, and between them there are...
Oh, between the islands themselves. I think that those timescales in this region, like it's in the order of millions of years.
But in Sundaland, in the other part.
Oh, yeah, I don't work there, but yeah, no, that's come up and down. I mean, for the silvereye, they haven't been there long enough. For some of the white eyes,
they may have been, but then they become single island endemics quite quickly. So it's definitely something to consider. I guess I haven't because I've been concentrating on the silvereye and all that stuff happened.
Yeah, I was thinking on a larger perspective.
Yeah.
Anybody else?
Hi, thanks so much for your talk. It was really interesting. I was wondering if you've done any genome-environment association analyses to look at this, differentiation that's not explained by chance. And if you have, or if you're going to, do you know what the environmental variables would be?
I haven't done it. I think one of them, so the work we do, we're trying to model the changes in biotic interactions that happen. That's one of the main things. I mean, these islands are different from an abiotic point of view in many different ways, but nothing in a repeatable way. But the way biotically they change is there's reduced predation, reduced inter-specific competition, because fewer things get there, and then the intra-specific competition goes through the roof because the populations get so big on these small islands. And so I think what we think is that it's the relative change in those types of biotic interactions that are driving the selection. But I haven't done we probably don't have, we could probably have a best guess, but we don't have all of the data on all those types of biotic interactions from all the islands. But what we're doing is using Heron Island, where we've just known every single bird since like 1968 or something like that. And we're actually focusing on getting comparative mainland data, because nobody was working on the mainland because it's really hard, because the chicks die, they get hammered by predation, and we're actually quantifying that now, so we should be able to do something like that in the nearish future. We probably need a couple of years of data to parameterize the models. Yeah.
Great, thanks.
Okay, thanks very much. We're out of time. Thanks, Sonya, again. And we'll see you downstairs at the Chiringuito.